Okay, we start.
Yeah.
For those who have ears and eyes present in the room and that want to hear. Okay, very good. I want to pick up where I left off yesterday. And what I was doing yesterday was first telling you some ideology about a kind of general problem in modern biology, namely the things you're interested in are not things for which you can submit CAD drawings to the great biological factory in the sky, but they're kind of probability distributions. You never, when you ask for, give me another human being, you don't get another BMW model 360.5. You get another human being who's clearly another human being, but drawn from some sort of random distribution. And we're happy with that, but it makes life rather difficult for people who want to think quantitatively about biology. So it's a kind of fundamental problem. And there is no general solution to it known to me. On the other hand, the way things are going, one knows how to collect data that's in quantities large enough so that you could begin thinking about trying to understand in some quantitative way the probability distributions of biological entities. And so what I talked about yesterday was an approach to the probability distribution on the sequences of the T cell and B cell receptors, the genes that confer recognition specificity on the central cells of the immune system. And I gave you a song and dance about how modern sequencing technology allows you to collect millions of examples from any individual human being of the sequence of the T cell receptor in the T cells that they have in their body. And we use this to basically to create a black box. This notional black box has as its claimed function to be able to generate sequences that are perfectly good sequences for T cell receptors in a human being. We can do it for any species we like, really. That, in a statistical sense, couldn't be distinguished from the ones that you would get by actually taking the sequences that are found in a real individual. So the idea was to generate what amounts to a null model, something that can be used to create ensembles of, in this case, sequences of T cell receptors that are statistically identical with those that are produced by the process that goes on in the stem cells that are producing the T cells and B cells in your body. And in order to do that, we had to inject some information into the system. In other words, we can't just take the examples that we've gotten and, in effect, take an empirical probability distribution. The entropy, how shall I put it, the diversity of sequences that could be produced is so large that none of the samples that we can ever take will really sample that distribution fully. So you have to have some kind of underlying model. And that was what I talked about mostly, was what is a sensible, sort of say, restricted model for how those sequences are generated, and can you infer the parameters that characterize that model from the data that you have. And the claim that I would make is that, yeah, we can do that. So, and now I want to talk today about some of the consequences of that story. All right, so I got you through to about here. I described the process by which you take the data that you can get from high throughput sequencing, what you do to infer from that data the parameters of your black box generative model. And then I showed some sample output. So this is sample output from analysis of human DNA sequences. And you find, as I said, the black box is characterized by a process in which a certain number of independent events occur. These independent events involve selecting of original gene modules from the chromosome that the process works on, deleting away some bases from ends of these modules, and randomly inserting bases in between pairs of modules that are brought together. So it's a modest number, 4 or 5, of independent stochastic events. And what you wind up extracting from the inference procedure is these elemental probability distributions that define the stochastic machine. And the simplest ones to think about are, well, what about insertions? I've brought together two gene modules, and I'm going to, in order to create my final gene product, I'm going to insert some number of bases in between. In first approximation, you might say, let's just assume that we insert bases randomly. That is to say, you insert A, C, T, or G with equal probability. Well, you'll find later on that actually there's a certain base bias. But it's not 25, 25, 25. It's a little bit different, but not so much different that it really changes things in a dramatic way. The one thing that is sort of dramatic is the number of bases that you get to insert is, on the average, something near 7 or 8, but it has a certain structure. There's sometimes you insert none, sometimes you insert 1, sometimes you insert 2. It's a rather odd kind of process, right? And so one of the things that comes out from the inference procedure, basically is the probability distribution of the number of bases that you insert each time you're constructing an insert. Okay, it has a certain structure to it. You could try to understand mechanistically what kind of crazy enzyme would do this, what's causing it. Obviously, it inserts a certain number, and then with some probability, it decides to stop, okay? You could characterize the process, if you like, in greater detail. It's a probabilistic process that could be described in greater detail. For our purposes, it hardly matters. Just to know what the stochastic output is good enough, and it would be interesting from the biochemical point of view to understand how it got to be that particular way, but I don't think it changes a heck of a lot in what I would say next. Okay, and so one of the other things that comes out is that it is significant, it is important that some number of bases are deleted away, and that deletion probability depends on the sequence that's being deleted, so it depends on which gene you're acting on, because these gene modules, they have different sequence. That's their whole point in some sense. And it's not unusual for enzymes that carry out some action on a DNA sequence to have sequence dependence. I mean, there are the restriction enzymes that you know from the bacterial world that choose a very particular motif, if you like, and cuts the DNA at that point. So the action of the enzyme is sequence dependent. This is a little bit more unusual. It's a rough and ready, not 100% probability sequence dependent action, but it's possible. It's easy to imagine how such a thing could come into being. And when I'm all done, I have the ability to characterize in a quantitative way just how diverse is the set of DNA sequences that characterize the T-cell receptor gene. Just how diverse is that set of sequences? You can calculate the entropy of the sequence distribution, okay? And so for purposes of advertisement and thought, it's worth knowing that the diversity is 47 bits, okay? That tells you that there's a rather large number of potential T-cell receptor sequences out there, and the number, it's interesting that it's large compared to the number of T-cell clones that you actually have in your body. All right, so if we were interested in quantifying the diversity, we can do it now. Okay, one thing that was I mentioned in passing, it's kind of interesting, is that if you can do this kind of thing for human beings, by the way, it's important that when you perform this inference process, it does not matter very much which human being you are using as your data source. The output of this process is supposed to be a characterization of some the action of some enzymes that carry out a process that every human being, unless they're really sick, carries out using essentially the same enzymes. So it should be something universal, and it in practice does seem to be. The other laboratory animal that you can readily work with is mouse. It's not the only one, but it's a very well-studied one, and you can readily get data. And on mouse, you can actually do a somewhat more elaborate experiment, namely, you can get sequence data from mice of different ages. A mature mouse, a mouse embryo, a mouse shortly after birth, a mouse somewhere between just born and mature. And it is kind of interesting because there you do see something actually happening. There's some dynamics. So you can perform the analysis that I mentioned before of figuring out what is the diversity of the TCR sequences that a mouse produces. It's substantially less than that of a human being. I can't read what it says up there. Oh yeah, so this is the same kind of presentation as that sequence diversity presentation for humans a few slides back. And basically, instead of 47 bits of diversity here, we have 30 bits. Okay, so that's 2 to the 10 is 1,000, so that's like 1,000 cubed, 10 to the 9 sequences instead of 10 to the 14. Okay? Mouse is a lot smaller animal. It has about 1,000th the volume of blood that we have, and therefore roughly 1,000th the number of total T cells. So it's perhaps reasonable that it should have a less diverse immune system. But the thing that I just wanted to point out is that if you perform this kind of analysis on blood drawn from let's see, what have we got here? Red is 6 week, 4 week, 6 day, 4 day. Okay, I think this actually may be mislabeled. I'm not 100% sure. What I've plotted here is the insertion distribution, the distribution on the number of insertions that are made between the gene modules that are brought together, and the analysis is done on mice of different ages. So these curves are the curves that describe basically what happens in a generic grown-up mouse. And a mouse is grown up from this point of view from, just a week or so old on. The other two curves are kind of amusing because the first curve, the one that is doing this, I think it's red, and it's just completely, it is mislabeled. That is for a, let's see, what is the correct, it is from a fetus that is a few days away from being born. And I forget exactly what the gestation period is, but you'll notice that it's very different in the sense that most of the time there are no insertions. There's a small probability of one insertion, and then the probability of higher numbers of insertions is dramatically lower. Just a couple of days after birth, this distribution looks a little bit lumpy. It looks, roughly speaking, like an equal superposition, if you will, of the fetal distribution and the adult distribution. So clearly there's some dynamics going on in the young mouse, and probably if there's some dynamics going on in the young mouse, there's some dynamics going on in the human, but you can't do that experiment. You wouldn't want to do it. And I think the biologists are not surprised by this. They know that the insertions are carried out by a particular enzyme called TDT, and for reasons that I don't understand, it turns out that the TDT enzyme is not made in the embryonic state. It is only made after birth. I guess the animal doesn't need its own immune system while it is still in the womb, and certainly needs it afterwards. But it's kind of amusing that you can see this phenomenon from this, so to say, looking at things through the lens of inferring the properties of the immune system. It also allows you to start playing some interesting games. Obviously, if the properties of this generative black box are changing with time, what you see when you collect the blood is going to be basically a superposition of distributions that were created at different times with different black boxes. So if you want to really understand what's going on, you have to be able to use the data that you've got to infer not just one black box, but what are the different black boxes at different times, et cetera, et cetera. It could be a nightmare, and I think it basically is, but it's an amusing challenge. All right, so I'm not going to go much further on that. I think this is where I got last time, and basically, so the end result of this procedure, the ability to construct the generative black box, is that for any sequence that you see, you can assign a probability of generation. That is to say, for that sequence, what's the probability that in a single generative stem cell event that sequence got produced as opposed to all of the other sequences that could be produced. Since there's such a large number of possible sequences, the probability of generating in one shot any particular sequence is, of course, going to be small, all right? On the other hand, it's a number, and now we have the ability to assign to any T-cell receptor that we observe that number, okay? And if I have a sample from a human being, I have, let's say, 100,000 sequences, I have 100,000 such numbers. These are theoretical numbers. There's not something that you actually observed in any direct way. You're using your model of the black box to, in fact, assign it. But it's an interesting distribution to look at. So these one-shot generation probabilities that you actually observe, that you assign to the actually observed sequences run from 10 to the minus 6 or 7, which is in fact a largish number when you think about the number of T cells that get generated to make a human repertoire. And it runs down to 10 to the minus 25. Occasionally you see somebody whose generation probability is 10 to the minus 30. You can make a histogram, okay? And indeed, and this blue curve is what the histogram of the sequences that come directly out of the generation mechanism looks like. This is the observed distribution, if you will. This is the distribution of the generative probability for the non-productive sequences. These are the guys who are in no way influenced by any other selective process. They're just directly representing what happens when the VDJ recombination takes place. Roughly speaking, if you take the average of this coordinate over this distribution, you get the entropy. That's the definition of the entropy. There is another curve here. The other curve is, what does this distribution of generative probabilities look like if I take the sequences that are productive? All right, so in my blood sample, as I explained, the sequences that are productive are sequences that are describing the actual presented T-cell receptors, and these are sequences that have not only been generated and at generation were, so to say, in frame, free of stop codons and all that stuff, and in addition, so they got made as proper surface expressed proteins, they made it through the thymus and got into the blood, and therefore are functional T-cell receptors. So not every productive sequence is going to make it through the thymus and into the blood as a potentially functional T-cell receptor, because we know that it's necessary to edit some of them away in order to avoid self reactions. All right, so the red and the green curves are this distribution of generative probability. All of these sequences, even though they have been selected, they're a sequence and they were made by this, this original generative probability, and they can be tagged with that probability. And so the distribution is interesting in the sense that the average has moved to the right. It's moved to higher generation probability. The green and the red are cells that are distinguished by being naive or memory. As I said before, it's possible to separate when you collect T cells from a blood sample on the basis of surface markers. You can distinguish between things that are naive, have never been activated, and cells that not only have been activated but have been passed on to the memory repertoire. And so this distribution is the same for both of those repertoires, and it surely means something that the selection process that takes place in going from they're made in the first place to, and they're in addition approved for service in your functioning immune system, selects for higher generation probabilities. So somehow the system is tuned to be better at making sequences. In other words, those, the stochastic machine with those parameters that we inferred by the process that I described is somehow tuned so that it's somewhat better at making sequences that will in fact pass thymic selection, all right? So the thymic selection is higher for the guys that are higher. The probability of passing thymic selection is higher for the sequences that are more readily produced. This surely means something, and it's an interesting hint, and it poses a question for evolution that, or for evolutionary thinking that may well be worth following up on. We do not know at this point exactly what to make of it. Okay, now, I so far have not told you anything that should reassure you that this number that I'm attaching to sequences, namely this generation probability, actually represents something that has physical reality. I haven't tested anything yet, okay? I've said, well, I can infer this generative mechanism. I can use it to assign a probability to sequences, but I haven't used this probability for anything just yet. Okay, but there is one thing that you can do, and that is, obviously, if we went back a micro, well, okay, obviously, if I have different individuals, since they are creating their own repertoires by independent generative processes, it might happen that exactly the same sequence got created in two different individuals. Well, you can look for that. That is to say, you can take your sample from individual A, your sample from individual B, and ask, are there any identical sequences, okay? Now, since these sequences are being produced by a chance process that is highly biased, that is to say, some sequences are much more likely to be produced than others, it had better be the case, if we have our hands on a correct estimation of the generative probabilities, it had better be the case that the shared sequences have much higher than normal probability of being generated. If that's not the case, then we're completely off track. So that's what this little analysis is about. Namely, if you take a collection of human sequences and you look for shared nucleotide sequences describing this region of the genome, and you ask, if I attribute this generation probability to these sequences, where do the shared sequences lie on this distribution of generative probability of all the sequences, all right? And so that's what this little blip here is. We had a collection of something like 7 human samples that had been analyzed in this way. Amongst those 7 samples, there were something like 21 shared sequences, and the red curve is the generative probability assigned, is the distribution of the generative probability assigned to those sequences, smoothed out a little bit. Clearly, they're coming from the high end of the generative probability curve. So qualitatively speaking, it's working the way you would expect. And you can go into this a little bit deeper, and you can ask for, how many sequences are shared. If you have, let's say, 7 samples, there will be some sequences that are shared between 2 of the samples. There are going to be a smaller number that are shared between 3 of the samples, and so on. And this is a curve of number of pairs of individual and number of shared sequences. And the blue is basically the predicted mean and variance, and the red are the actual numbers. So, we're doing pretty well. This is for, and of course, the numbers that you expect here are highly influenced by just how big your samples are. All right, the bigger the sample, the more opportunities for chance coincidence that you have, and you should have more. So this whole analysis, of course, is predicated on keeping careful track of sample size. Okay, so this is just to say that at least at this level, the probabilities that we have been talking about would appear to have some objective reality. They have an impact on something that depends upon real physical probability. Okay, as a further little bit of information that I won't go into more than briefly, this whole exercise so far has been carried out on T cells. Can't live without T cells. You can't live without B cells either. They're a different beast. On the other hand, their diversity is generated by essentially the same process. The same enzymes are just working on cells that have gone onto a different branch in the diversification of the hematopoietic lineage. So for B cells, the most important thing to note is that the distribution of insertions, this thing that generates, that is responsible for the largest fraction of the entropy of the sequence distribution of T cells, is much broader. In other words, the mean number of insertions is substantially larger for B cells than for T cells. And if you, follow the line, if you follow that observation down the line, you discover that the net effect on sequence entropy of the B cell surface proteins is that instead of being 47 bits, the entropy is 74 bits. It's really quite remarkably larger. What did I say? 47, so it's 30, 25 bits more or something like that. That's really quite a lot. If you look at where the entropy comes from, actually it turns out the entropy from gene choice, the entropy from deletions, the entropy from gene choice, the entropy from deletions, if you go back and compare it with what you get for the T cells, not much different. The big difference comes from just having much longer insertions. Again, why does the B cell lineage need all of this diversity? That is not clear. It is a lot. It certainly feels like overkill. Probably doesn't cost the organism much to do this, but it must have settled on doing it this way for some reason. All right, so anyway, B cells have their own story to tell. And okay, all right. We now have to dig into a much harder problem, and that is, what can we say about the productive sequences? They have been selected in some fashion. Okay, now when you come to the productive sequences, well, these are sequences that can be translated into sequences of amino acids and therefore turned into proteins. Now, it is generally thought, and it may even be actually true, that, well, it's certainly got to be the case that what matters in the end for function is not what the nucleotide sequence of some protein is, but what its amino acid sequence is. So that raises an interesting theoretical issue right away, namely, what I've constructed for you so far is the probability of generating a particular nucleotide sequence. Now, in reality, what matters is the amino acid sequence. So what I really should give you for any question that I ask is, what's the probability of generating a particular amino acid sequence? Well, That means that I have to be able to, in effect, sum over all of the probabilities of generating all of the nucleotide sequences that give you a particular set of amino acids. Because of the degeneracy of the genetic code, and because we're talking about things that are some non-trivial number of residues long, that ambiguity becomes very, very, very large. Okay, so there's a technical problem. If you want to know a different number, not what's the probability of generating a nucleotide sequence, but what's the probability of generating an amino acid sequence, you have to perform another level of computation or analysis or cleverness. And so I can't tell you the details because it's just too painful to explain. However, the way this whole problem is set up, the first thing that you discover is, okay, I can figure out how to get the generation probability of an individual nucleotide sequence. In principle, I could then march through every last nucleotide sequence that is responsible for a particular amino acid sequence and sum up those probabilities, and that would be a correct answer. That is just computationally impractical, but it turns out that the way the whole process works, it's set up in such a way that a technique called dynamic programming basically allows you to short-circuit this awful, so to say, combinatorially exploding calculational problem, and you can turn the whole problem into something that is quite manageable. So I won't tell you the details, but there is an improvement of the method of calculation that basically allows you, once you've figured out what the stochastic machinery is for generating nucleotide sequences, there's a way of organizing the computation so that it becomes possible and even quite efficient to calculate the net generation probability of something where you specify the amino acid sequence. All right, so that actually was a lot of work, but it was worth doing. Now, in addition, I showed you what a typical sequence for the protein, for the surface receptor. Yes?
No. You're telling us about productive or non-productive services?
Well, at this point, since we're talking about I have a productive sequence.
Okay.
It has an amino acid sequence, and I want to know what's the probability of generating that guy. This is, now, again, I'm saying what was the probability that my stem cell rearrangement event produced one of the nucleotide sequences that does give you that amino acid sequence. So it's the primitive generation probability, not of a specific nucleotide sequence, but of a specific amino acid sequence, okay? I haven't yet worried about, given that I've got this amino acid sequence produced at the stem cell level, what happens as it passes through the thymus? I haven't talked about that yet. But, in order to be presented to the thymus, I have to be generated, okay? So knowing the generation probability of an amino acid sequence is definitely worth having. In addition, yeah?
Are you working with the whole gene only with the exons?
Okay, sorry, only the?
Exons.
Well, we're only working with the, yeah. We're only working with the exon. And in the end, we're going to accept the biological wisdom, maybe not shared by all biologists, but I think it's generally accepted, namely that the part of the gene that really matters is this restricted region of maybe 30 residues long called the CDR3, complementarity defining region number 3. That's the one that actually gets in contact with the presented peptide. All right, so this is an approximation, but I'm going to talk to you about the generative probabilities of amino acid sequences that are restricted to this CDR3 region. okay? This is a helpful convenience. The flanking regions of the gene are not unimportant, but they don't contact the peptide that's presented. But they do contact the MHC molecule that's presenting the peptide, and therefore they will have an impact on the overall functioning of the system. But we're just trying to get our feet wet and understand in some very global terms how things are working, okay? All right, but anyway, what I wanted to focus on at this point was these probabilities are very nice, but I have to step up my game and figure out probabilities for things, for genes defined by amino acid sequence, all right? Okay, so, and this is what I want to show you here. So if I give you an amino acid sequence, obviously it's a lot easier, it has a lot higher generation probability than that of any one of the nucleotide sequences that it is equivalent to, okay? And the question is, how big an effect is that? Because these probability numbers are very small, and we're going to talk about things that have to do, how likely is it that you're going to see a T-cell receptor that does some particular job? How likely is it that that T-cell receptor gets generated? Well, what I really want to know is how likely is it to be generated as an amino acid sequence. So I have to know how much of a boost do my probabilities get when I pass from nucleotide to amino acid sequence. It's a trivial thing, but you have to do it. It's bookkeeping. All right, so here's the effect of the bookkeeping. So for the sequences we've been talking about mostly are for human so-called beta chain of the T-cell receptor. There's a slight annoyance that there are two chains, two proteins. One is called the alpha, and one is called the beta. The alpha chain is longer. It's thought to have a larger impact on the specificity, but the alpha chain with which it combines to make a heterodimer is essential for the protein that gets expressed on the surface, and it's also essential for the functioning, except if you're a llama. If you're a llama, you only have a beta chain. You don't have an alpha chain. So apparently there are some animals that can do without it. So if we wanted to do llama medicine, if we were in the veterinary school, I think we would be in a much better theoretical position than if we want to participate in activities in a human medical school. This is unfortunate, but this is the way it is. All right, anyhow, back to bookkeeping, which we have a better control over. So focus on the red and the blue curve. The red curve is the same thing that I showed you before, namely, it's the distribution of primitive generation probabilities of the productive sequences, the productive nucleotide sequences that you see in a human sample, all right? The blue is what happens if basically you add together all of the sequences, all of the nucleotide sequences that gave a particular amino acid sequence. And what you see is that, so obviously, every probability is going to move to the right. And if you do that collectively, you see that the probability distribution moves to the right quite a lot, all right? So the blue curve peak is now much closer to, so to say, the max than the red curve. So many nucleotides give the same CDR3 amino acid sequence. I call this a coarse graining. And this coarse graining, namely putting a lot of initially different things into the same category, all right? So we put many, many different nucleotide sequences into the same pot where they all correspond to the same amino acid sequence. And that causes the probability distribution to shift rather impressively to the right. I mean, the mean has gone up by something like a factor of 1,000. Not bad. All right. So each human has something like 10 to the 9 clones in their body. In other words, you've got, we don't know exactly what this number is. We know exactly, or well enough, what the total number of T cells is. Let's call it 10 to the 10-ish. And we don't have a good handle on what's the distribution of the clone sizes. In other words, a given T cell sequence can be found not just in one T cell, but in a bunch of T cells. And the bunch of T cells that have the same sequence is called a clone, and there is some distribution of clone sizes. And we haven't talked about clone sizes at all yet, and probably won't today. So estimates of how many clones, how many different sequences you have in your body is about 10 to the 9. Thus, any clone whose generative probability is greater than 10 to the minus 9, now we're talking about humans, is pretty likely to appear at least once. I mean, I can turn this into a precise probability statement, but roughly speaking, 1 over the number of clones in your body is the borderline between everybody in the human race is going to have one of these, and very few people in the human race. Well, now we have to talk about it in more detail, but the probabilistic issue becomes more complicated. But there is a line you can draw on this curve, let's put it at roughly 10 to the minus 9, where anybody to the right of that has a high enough probability of generation that everybody in the human race is likely to have it. Okay, so there's a significant fraction of our T cells, if we look at them through the lens of amino acid sequence, as we should, that everybody has. And obviously, evolution has done something to select this collection of sequences. Now, the thing that I find really quite fascinating is, okay, so this fact means that in human repertoires, many clones will be what the immunologists call public. Everybody's got them. But this is just by chance statistics of VDJ recombination. One is likely to say, well, if everybody has this thing, it must be because it's useful. It has some functional utility. Maybe. It's also the case that they're there because of the way the stochastic machine is structured and the way the genetic code is structured and so on. Maybe the two are the same thing, but conceptually speaking, sharing because of chance probability and sharing because of evolution drove that particular sequence into everybody because there is some species-wide selective advantage. They're not the same thing. Okay, what is amusing is what happens for mouse. Mouse is smaller by about a factor of 1,000 in size, number of cells in their immune system. And let's see, for mouse then we decided that the dividing line is probably p-gen of order 10 to the minus 7. And interestingly enough, for mouse, the distribution is already higher, towards higher probability because of the way the generative machine is structured. And then when you do this further coarse graining onto amino acid, the probability distribution shifts further to the right. And interestingly enough, the transition between everybody's likely to have this amino acid sequence and it'll be not shared by everybody is, roughly somewhere in the middle of the distribution. So it's not obvious why mouse has to have less diversity because it lives in the same world we do. And it's not obvious why things should have adjusted themselves in this way. I'll come back to this point a little bit more in a second. Okay, a statistical view of CDR3 amino acid sequences. Well, it's worth perhaps looking at this. CDR3, the active region, the CDR3 region of the T cell receptor gene is of variable length. So you have a little bit of a technical problem if you want to compare sequences and ask about gene utilization. But you can actually rather nicely align the sequences of different length because they have beginning and ending regions which are quite characteristic, and alignment with gaps allows you to line up the sequences of different length. And you can see that there's a very characteristic sequence logo of amino acid use. Everybody starts with a cysteine and ends with a phenylalanine, and then as you go further in, you see you see more entropy, but nowhere near maximum entropy, but getting more entropic as you go towards the interior of the sequence. And this is simply reflecting the fact that towards the left end and the right end, you're really seeing the V gene or the J gene, and they are, they have restricted variability, and you start seeing really random stuff in the middle, and various things could be said about this. So it's not a random collection of amino acid sequences by any means. I guess I'm not going to have time to actually go into any depth into this, but there's a subject that I have avoided talking about because it's complex, but it has to be faced at some point, which is that all of these probabilities that I've talked about are probabilities of generating a sequence, so to say, in the stem cell event. Has nothing to do, did not yet have any way of telling you about what's the probability once it's generated that it makes its way into the active population of T cells, passive thymic selection, let's say. Well, you can do something about that, and we have. Let's see, T cells once generated have to both fold and then they have to pass thymic selection before they can pass into the blood. Okay, so we'd like to model the statistics of real CDR3s, and if we want to do that, we have to add selection effects to the generative model. Okay, two options exist. Okay, there's an easy way and there's a hard way. Let me just briefly summarize the hard way. The hard way is, presumably there's some sequence dependence of the selection process. So you really have no choice, just as we had no choice in trying to figure out what was the primitive generation process, we had to have some model. We had to have some restricted model for how the diversity is generated. But at that level, we had lots of biological information. We didn't have to make a bioinformatic guess. We could say, we know what the enzymes that do the recombination do, functionally speaking. We don't know the details, but we know what happens. You choose a gene module, you make insertions, you make deletions, and you just have to describe those things independently. When selection comes into the game, it's much more complicated. We don't know. So you have to make a model, and you could make a model and then see whether it works. And the thing that we have tried and does seem to work in a significant way is essentially to imagine that the probability that a sequence gets selected once it's generated is basically a product of factors that reflect what is the amino acid sequence at different positions within the CDR3. In other words, just assume that the probability is a set of is the product of independent probabilities for selection computed on a position by position basis. And there are a few further decorations of that you might want to think about. So that's the hard way, building a model that captures sequence and CDR3 position-dependent relative selection effects. And we can fit a simple multiplicative model to the data, and we adjust the, so there must be some other, yeah, there we go. So the simplified model that you could try out basically is that the ratio of the probability of generating to the probability of both generating and passing selection is a factor which depends on the sequence, and it depends on the sequence by a product of factors that take into account which position in the sequence you are in the CDR3 and which amino acid you are. And then you do an inference procedure of a kind not unlike what I described before. It's pretty complicated, but the remarkable thing is that it works reasonably well. Or I'd say it works quite well. And you can infer all of these factors in this model for the ratio between the selection probability and the generation probability. And it's very stable. You get the same answers from different individuals. You get the same answers if you ask for, well, I won't go into this in detail. It's hard work, but it can be done, and it seems to make a certain amount of sense. Now there's an easy way, which may sound to you crazy, but it's worth thinking about, and that's what I would like to show at the very end here. The easy way is the following. When generating a synthetic repertoire, accept a random fraction of the generated sequences. So our goal here, in the end, is to create a machinery that will allow me to construct a null model. That is to say, a distribution on sequences that basically contains all of the essential selective and generative phenomena that characterize the generation of a human immune system, okay? The reason why we want to have this is that we would like to now use this in the case of, this is not a generic human, this is a human that has a disease or some such thing, and we would like to see the small changes in the distribution that reflect this anomalous situation of this particular individual. But you have to know what the background looks like. You have to understand the background sequence distribution in order to see small effects. So our goal is to generate, excuse me, a null model for a working human immune system. So our easy way for dealing with selection is what I will now describe, and then I'll show you a consequence of accepting the easy way and argue that, well, maybe not a bad idea. So in order to create my null model, I generate synthetic immune systems. I generate hundreds of thousands of examples from my generative procedure. Now, how am I going to model selection? Well, I'm going to model selection by saying that I don't understand why a particular amino acid sequence is selected for going from generation to actual incorporation in the immune system. Suppose I make the assumption that it's just random. That is to say, some fraction of the generated sequences are accepted, presumably a relatively small fraction, 10%, 5%, who knows, something in that general area seems reasonable, but it's random. So I'll figure out how to choose my random selection probability later on, but the idea is to construct my null model, I generate sequences using the methodology I've already described. At random, I throw stuff away. Maybe I throw away 90% of what I generated. But the idea is, having generated a sequence that I throw away, I'm saying that that particular amino acid sequence is for some reason bad. And if I ever generate it again, I throw it away again, okay? So it's not only bad for me, but it's bad for anybody else. It doesn't fold as a protein, or it has some other feature that makes it undesirable. So you can generate a null model for selected sequences by this process that I've just described. I hope it's a little bit slippery, but I hope it's clear what I'm doing, all right? I'm generating them using the generative model, which I think is pretty clear, and then I'm throwing away, or selecting, if you want to talk about the inverse, I'm throwing away sequences at random, but once I've decided that a particular particular amino acid sequence does not pass thymic selection, it gets thrown away. It doesn't, even if I generate it again, it doesn't pass thymic selection, it's just bad. And there are many reasons why a particular sequence could be bad. All right, so what happens if I do that? What happens if I do that? All right. So if you do that, you can generate T-cell repertoires of any size. And you can test whether the real and synthetic statistics agree. For example, once again, we can come back to the issue of sharing of sequences between different individuals. We came across a really interesting data set. For some reason, one of the research groups that works in this area had generated T-cell receptor data sets for 658 ordinary, randomly chosen, healthy human beings from a range of ages, with roughly speaking 40,000 unique nucleotide sequences for each of them. And the total number of distinct amino acid sequences in the whole database was like 30 million, and they're divided up handily for you into 658 pots corresponding to 658 human beings. And you can, of course, immediately ask, well, you know, are there any amino acid sequences that are shared between individuals? Are there any amino acids that are shared between all 658? There shouldn't be very many. Are there other amino acids that are shared between 500 of the 658, et cetera? In other words, you could ask for empirical sharing of amino acid sequences between between varying numbers of individuals in this large data set. We can, according to our model, we can create synthetic data sets of the same sizes for 658 synthetic individuals. We can do this over and over again if we're so inclined. And we can ask for our synthetic data sets. Is there sharing of sequences between individuals? In order to create our synthetic data sets, we had to make a choice about this probability of accepting something that I generate, or probability of throwing away things that I generate. There's a completely orthogonal way of deciding what that throwaway probability must be. And let's see if I can explain that. But let's just say there's an independent method that has nothing to do with sharing statistics that allows me to decide what is a good choice of this selection probability. How many sequences do I throw away because they don't pass selection? And so I make that choice, and it's indicated here. Let's see. Right. Let me just say a word about how I make this choice. So the probability of keeping a sequence in my process of generating my null model turns out to be 1 in 25, 0.04, all right? I fit that to a completely different story. That is to say, in the real data, you have a certain number of nucleotide sequences and a certain number of amino acid sequences. Many nucleotide sequences will map onto the same amino acid sequence. And as you take more and more nucleotide sequences in, you get more and more different amino acid sequences. However, the rate of increase is not 1, right? And so one thing that you can do is to choose your throwaway probability so that you match the actual statistics of how does the number of distinct amino acid sequences increase as the number of distinct nucleotide sequences in your data set increase. So that's what this is about. This is, as I increase the number of unique nucleotide reads, the number of CDR3 reads increases, and the red line is what I get from my generative model with throwaway factor Q is equal to 0.04. And the blue crosses basically are take the real data set and ask the same question for different numbers of sequences chosen from the, picked out of the real data set. So I fix this throwaway factor by a particular matching of the data to the model, but then I ask the question about sharing. That is to say, in my now I can generate my synthetic repertoire of human beings, 658 human beings, each one of which has a sample of whatever the sample number is. But it's important to match the sample sizes because sharing really does depend on how big your sample size is, so you're not getting a good test unless you match the sample sizes as well. But we do that. And then, you know, the number of CDR3s that are unique, they have nobody shares them, is some number times 10 to the 7 because we have a few times 10 to the 7 sequences in total. The number that are shared with one other, the shared between two pots, between two individuals, is less, and the number shared amongst 3 is less, et cetera, et cetera, et cetera. So the crosses, of course, are the data. What you actually find when you take this set of 657 individuals, and the red line is the model constructed as I've described it. And it is quite remarkable. You're looking at relatively high powers of the, how shall I put it, high order correlation functions of the probability distribution when you're looking at high sharing numbers. But obviously this simple method of generating a selected repertoire is, at least as far as sharing probability goes, which is a pretty sensitive test, doing a remarkably good job. We should tests that are more detailed and, I don't know, more precisely data-driven selection model does as well as this, or possibly better. But in any case, what I wanted to say here is that you have to do something about how the distribution of sequences is modified by selection. And I'm trying to tell you that there are things that you can do that appear to be plausible, appear to agree with what actually happens well enough so that you could perhaps take this to the next step, whatever that next step is. Okay, there's a lot of fun stuff that you could go through. You can analytically compute all of these sharing statistics if you're so inclined. You learn a lot about how the whole thing works when you do this, but don't have time to do that. Yes, okay. Yeah, so there are two final things I want to say before I stop. Let's see, do I want to say this? Okay, we can evaluate the fraction of the T-cell beta chain repertoire that will be public in the sense of, right, repertoire size, percent, okay. So, okay, what we said is if your repertoire size is n, having generation probability bigger than 1 over n is having a sequence with generation probability greater than 1 over n, that's a sequence that has a high probability of being shared with other individuals, okay? And therefore public in the sense of immunology. Now, let's see. We can compute, right, and so the question therefore is, what fraction of the repertoire, you know, we have these histograms of generation probability. And for such a histogram, you can compute what fraction of the repertoire lies above any given generation probability that you choose, all right? So I can plot for any species where I have this distribution, I can plot what percent of the repertoire has generation probability bigger than 1 over n for different n's, okay? I know what the size, I know what n is, let's say, for the human repertoire, but, you know, we don't know why it's what it is. We don't know why it's 10 to the 9 or 10 to the 10. It could have been 10 to the 12. Well, not really. We'd be an elephant if it were 10 to the 12. But we could have had a sparser repertoire. So in any case, given the distribution that I've computed, I can plot as a function of repertoire size what percent of the repertoire will lie between, will be greater than 1 over n, where n is down here. I should have said repertoire size n. So for humans, you see a curve like this. For mouse, you see a curve like this, and the red bar is the rough range of our estimates of how many different clones there are in mouse, and the blue bar is our rough range of how many clones there are in the human being. And the thing that is interesting, and you have to process this in your head a little bit before it becomes intuitive, but what's interesting is the fact that, well, the fraction of the repertoire, for the actual range of human repertoire sizes, the fraction of the repertoire that is going to be public in this sense is therefore indicated by this bar. Somewhere around 15%. That's sizable, right? Because given that's a lot of different sequences. And for mouse, it's even bigger, but it's around 30%. But again, it's different from human. It happens to be bigger than human, but the point of interest is, despite the enormous diversity of the immune system, the fraction of our immune system, or the fraction of the mouse system, that is shared between all individuals of the species is, order 1 at the one significant figure level, 15% or 30%. It's not 0.001. It could easily have been 0.001, all right? So I find this intriguing, and I think the thing that obviously somebody should do, maybe I'll find an undergraduate who's willing to do this. Obviously, you want to put a few other species on here. You want to have elephant, you want to have trout, you want to have, name some other intermediate size animal, dog. Well, dogs are too variable in size. So whether this is just an accident or whether this is something that is of larger significance, I don't know. Yeah, well, you can say the human race hasn't fully sampled TCR space. That is an important fact, and I will. Okay, I'm going to close with the following thoughts.
Sorry, can I ask a question about this public repertoire?
Yes.
Say, for example, when I don't know whether this is relevant for transplant, where people want to check for compatibility of immune
Right.
system. So in that case, I think there is a part which depends on the sequence. And a part, if you take two different individuals, probably they will not have the same BDJ. There will be mutations and probably this diversity.
Well, yes, these modules that are joined together to make the recombined gene,
Yeah.
they do have alleles.
Yeah, so the question is, say, this 15% should depend on whether
Well, actually,
comparing a relative to
yeah.
completely different individual.
I'm completely glossing over that question. So I'm talking here about sharing of the CDR3 region of the gene, not worrying about flanking regions which might have alleles in them. That also will be important for having control of the effect of, HLA type, the different MHC molecules. That part of the story gets more complicated, for sure, all right? And I don't master it. It's one of the things that I'm gradually learning how to understand.
To some extent, in your model, the sequence is the same. And you are matching data where the sequence might not be the same.
Possible.
Maybe it is 650.
Yeah. I mean, I'm not even, the HLA type might even be known, but we don't worry about it. Because we're only focusing on this small, so to say, thing that's important for peptide MHC recognition in the sense of directly recognizing the peptide. How efficiently you recognize the peptide presented on the patient's particular MHC class, that's another matter, which I haven't gotten into, all right? Okay, last remark. The flip side of the fact that a large fraction of the total repertoire is public is that a large fraction will be private, that is to say, shared with next to nobody. And you might ask, whether these sequences that are private, not shared with anybody, are good for anything. all right? Do we have them for a reason, or are they just, I mean, how shall I put it? Are they functionally useful, or is it the case that they're there because this kind of generative process simply inevitably creates them, and there's nothing you can do to stop that? Okay, so here's a moderately interesting way of getting insight into this. There are large databases of CDR3 sequences, T-cell sequences, that are known to be responsive to various diseases, or even responsive to various epitopes of diseases. Exactly how these data are collected is not terribly well curated or known, but if you just take these databases at face value and simply ask, okay, let's pick, I don't know, pick yellow fever, or influenza A, or hepatitis C. For each of these diseases, somebody has nicely gone through all of the literature and put together all of the experiments where somebody took the appropriate epitope of yellow fever and fished in blood of human beings and found which T-cell sequences respond to that particular epitope. So you get epitope associated with the disease, or in fact, all epitopes associated with the disease, and T-cell sequences that have been observed to respond in some fashion to that epitope, maybe with tetramer assays or something like that. So these are typically lists of sequences that are a few thousand long, all right? Long enough so that you can do some amusing statistics. And you can ask, so take those T-cell sequences which have been certified to actually play a role in responding to an actual disease. How does their generative probability lie on the scale that we have learned to understand? And it turns out that very nicely, the responsive epitopes follow they follow the distribution of generative probabilities that our repertoires as a whole follow. In other words, they are not concentrated at the high end of the probability distribution. It's impossible to distinguish these distributions from the background human distribution. So the guys that are lying up here are going to be public, but the guys that are down here are not public, and yet they clearly are responsive to a disease, and therefore they're useful. Okay, exactly how we play this game and exactly what we learn from this from the point of view of fundamental science and medicine remains to be seen. But in any case, private CDR3 sequences are functionally important, all right? So the public repertoire is there, the private repertoire is there. Response to disease, which is what the immune system is supposed to do, clearly straddles the boundary. And so that's good to know, something that you're going to have to work with. And one of the reasons why I'm interested in this game is because of immunotherapy. And the one sentence or two sentence explanation of why it's relevant is that, well, any given cancer sufferer has got a particular set of mutations of its own genes that is responsible for the tumor, for the cancerous behavior. And in principle, its immune system ought to be able to recognize and deal with the cells that carry those mutations. That's what the immune system is supposed to be for. Immunotherapy drugs block the ability of tumor cells to turn off the immune system, which is typically what they do. So if you unleash the immune system, why doesn't it go to work on the cancer cells and cure the patient? It does in some cases, but in many, many cases it doesn't. What's going on? Well, there's a statistical question there. In other words, for the immunotherapy to work, the patient's immune system has got to have the right set of T-cell receptors to respond to the mutations that are responsible for the cancer, or mutations in the cancer cell. Whether or not that's something that will generically be true depends on the statistics of the mutations, the statistics of the T cell, the statistics of how mutated peptides and T cells interact with one another. So there's a whole bunch of statistical questions that lie behind the potential for immunotherapy as an actual useful therapy for human health. And it's kind of interesting that this practical question intersects, it seems to me, at a fundamental level with some really interesting fundamental questions in the statistics of the immune system. So that's why I feel it's a really important subject area to explore, and I think there's still room for considerable advances. Lots of new kinds of data are being collected, and there's just a raft of conceptual questions that can be addressed. And so I think it's a really interesting area to pursue, and it's, I guess, icing on the cake is that you can see clearly how advances in fundamental understanding might have a practical impact in medicine, and therefore, you can feel good about thinking about it, not wasting your time. Okay, it's been a pleasure, and maybe I should offer to answer a few questions. I have to leave, I would say, in 15 minutes. So if we're going to do questions, we should do it now. And I tried to say far too much in too short a time, and therefore, if anybody's really interested in this stuff, they surely have questions.
So are there any questions? Yeah.
So what about, you show that B cells have a much wider variability. So is there a public repertoire also for B cells?
Well, I guess everything that I've said maps onto B cells, right? There's a CDR3 region, it's longer and therefore much more variable. You would expect public to be a much more restrictive, how shall I put it, criterion. And the answer is, that's a very good question. I just haven't had time to think about it. I don't know. I'm going to guess that I'm baffled by why B cells are so variable, but it's an extremely interesting question, or rather, it may or may not be interesting, but it's one that should be clarified, and it should be straightforward to clarify it. I just haven't done it.
I think I have a question. There is one there. Okay.
You said something about the pattern in the graphs of cells, human cells and rat cells, and I was curious about it. So I don't know if you had the time to explain it.
Okay, so the core of the question was what? It was about?
Yeah, the comparison between the cells, human cells and mouse cells.
Between mouse and human?
Yeah, the graph that you said was a pattern in the how they were placed together, and I think you said that had a curious thing about it, so I was curious.
This is the comparison between human and mice?
Yeah, that's right. Sorry.
But in what respect? And the
is in the graph.
Yes.
About the red line. I don't remember exactly how it is, the graph. Sorry.
This one?
No.
Further back.
The back.
All right.
Yeah.
Here.
Yeah.
Ah, okay. All right. So, this is simply to make evident one thing. In other words, for human and for mouse, the red curve is the histogram of generative probability for, a typical mouse collection of cells, a sample or the whole blood capacity of the mouse or whatever. The blue curve is what happens if you combine together all nucleotide sequences that have the same CDR3 amino acid sequence. So it's a coarse graining, if you like, of this distribution, and because there are many different nucleotide sequences that give you a particular amino acid sequence, and you're supposed to add up all of the probabilities to get the probability of generating an amino acid sequence, everybody moves to the right. So here, the red curve has a mean indicated by that dashed line, and the blue curve has a mean indicated by the black dashed line, rather the blue dashed line. Roughly speaking, the generative probabilities sort of per a particular class of sequence move up by about a factor of 1,000. All right. So, and this is just to say the same thing happens for mouse. The distribution of generation probabilities is initially much further towards 1, right? Much further to the right for mouse, for reasons, well, I'm not sure exactly why functionally that is the case, but it just is the case. The mouse, the mouse genome is different. The number of insertions that mouse seems to want to make is smaller. So the generation probability of a typical mouse TCR sequence is to start with higher anyway, and then when you put things together to make the generation probability of an amino acid sequence, it moves really quite far to the right. So, that's the blue curve here, and the mean generation probability is somewhere around 10 to the minus 7. Mm-hmm. It's just a fact that the number of clones that a mouse has in its body is like 10 to the 7. So, some substantial fraction of its repertoire has a probability of generation that is higher than this magical 1 over the size of the repertoire number. And so, the actual sequences that lie here under the blue curve, they are sequences that you're likely to find in mouse 1, mouse 2, mouse 3, et cetera. And so the same thing is true for human. And the question that I came to a few slides further on was, well, what fraction of the total repertoire lies to the right of 1 over n when I choose n to be the actual size of the repertoire? See, in all of these plots, there's nothing is said about how many total sequences I'm working with, right? The repertoire size is totally up for grabs, but now it is what it is. And why it is what it is has to do with, well, how big the animal is for starters, but it's not the only thing.
Thanks.
Did that go in the right direction?
I understood that the comparison is it would be need to compare it with other animals to see the exact relation between these?
No, it clearly gets to be interesting to look at other species. And so in order, it has to be the case that somebody has found it interesting to do this kind of T-cell sequencing experiment on other species. So we know that people have done it for fish. Fish are very important because we eat them. We eat cows too, but I don't think anybody's done a serious study of cow immunology. And beyond that, I don't know. I mean, if it becomes interesting, then it's a trivial matter to get this kind of data. It's actually really cheap, Mm-hmm. I think you could go to the butcher shop and get a perfectly adequate sample of cow blood to send off to one of these sequencing houses and actually get data that you could use for a study like this. It's really weird. Somebody just has to think it's worth spending a few $1,000s.
So maybe going to the 26th slide, if you may, please.
You want me to advance the slide?
26.
Wow. Okay.
Okay. So it's a remarkable match, right? And it sort of fails towards the end, right? So
Well, yes, right. I mean, let me go back to somewhere way back here. Here, this way. Okay, so if you look at the equation up top, when you're asking what is the probability that randomly generated samples of certain sizes will have the same sequence in them, okay? What you're doing is performing a kind of correlation sum on the probability distribution. So you notice here the probability of finding pairs. So if I have two human distributions and I know the p-gen distribution and I want to know if I take N1 samples from human 1 and N2 samples from human 2, and I want to know what is the predicted number of identical sequences in those two guys. Well, what this says is that the answer is going to be the product of the sample sizes times this quantity, which is basically the sum over all possible sequences of the product of their probabilities of being generated. So roughly, it is an integral over the square of the probability distribution that tells you the probability of finding pairs. If you wanted to know, if you had three samples and you wanted to know what's the probability or what's the mean number of cases of somebody that's present in all three, you'll be doing a sum of the cube of the probability distribution, and so on and so on and so on. So if we go back to 26, what you're doing when you're looking at sharing numbers, that are getting into the tens or the hundreds, you're implicitly doing some kind of convolution of the probability distribution to a very high order. And, I'm astounded that it works as well as it does, that I start to deviate, down here at sharing numbers of hundreds. That does not make me feel unhappy at all, because that's asking such a precise question of the probability distribution that it's remarkable. I mean, it's only wrong by a factor of 10 out here or something like that. That's amazing. So I'm a little bit embarrassed that it's this close. I'm embarrassed and surprised that it sort of lies within the error bars out to sharing numbers of many tens, right? So it would be, how shall I put it? You should have said, I don't believe you if you saw me agreeing all the way out to the end. I almost don't believe myself.
Right. But for following that reasoning of such a convolution, it would seem that you should probably fail. Again, it's not a failure.
Right. So when you ask them a more sensitive question, the likelihood that you have, think a probability distribution, right? Probability mass in some space, it'll have tails out in various directions. And these tails may be Gaussian tails, or they may be long tails, fat tails. And there are questions that you can ask that basically are heavily weighted by what's going on in the tails of the probability distribution. And, the likelihood that you get all the tails of the probability distribution right when you're constructing a model goes down as you're asking more and more sensitive questions.
Right. So maybe the data should be up the model.
Or you're asking why the data is fair question. I don't know the answer to that at the moment. All right. You want to know why my model predicts more sharing than the data actually shows. Okay, fair enough. Out at 100. That is a interesting question, and you'd have to really get your hands on the details, the analytic details of the probability distribution to answer it. So, fine question, and I can't answer on my feet. I don't know. All right, I would I would guide your eye towards the rather long stretch where the model and the data actually seem to agree remarkably well. I'm surprised by that already because it's asking a very sensitive question.
Okay, so I think we should thank Curtis again. Thank you very much for being here. It has been a pleasure to have you here.
Okay. All right. Time for coffee, yes?
Oh, yeah. So we have coffee now, and I think we are back at 11:20. 11:30, yeah.